Thanks a lot. Hi, everyone. It's a pleasure to be here. Thanks a lot to all the organizers for this fantastic meeting. Been enjoying the science, also the sun, at least yesterday. Today is a bit less. So I've got a picture of that, of the sun yesterday. Also, the sunset was great. And I have to say, these EMBL meetings, they're just getting hotter and hotter each year. So yesterday evening, I was looking online about the weather throughout the last 50 years here at Heidelberg. So this is specifically for Heidelberg. The blue color is cooler weather, and the red is warmer weather. And so you can see the trend pretty clearly, I think. And I'm sure that you all agree that this is not so special. We know that this is happening all over the world. There are rising temperatures, and this is changing ecosystems. It's shifting habitats. It's providing, it's leading to more and more droughts, rain, fires, you name it, right? Apart from all these environmental changes that I just mentioned, they strongly affect animal physiology, behavior, and their survival. And it's not just limited to the classical polar bears, of course. So in our lab, we mainly look at frogs and fish, ectotherms. So they use the environment for their body heat. And especially their embryos are also very vulnerable to environmental changes. And so these animals, they are sensitive to their environment, as I just explained, but not just their external environment, but they also sense the temperature internally, right? So they're controlled by biochemical reaction networks that are all over for their regulatory networks to regulate their behavior and all their processes. And so if we look at these biochemical networks and how temperature affects that, you've got each reaction that could, in principle, be affected differently by temperature. And at the microscopic level, then, you've got particles that, when temperature increases, start moving more rapidly, and typically that increases the reaction rates. And so overall, this affects all of these reactions, but the trick is, as I just said, or the thing is that they can all be affected in a different way. They can have different sensitivities to temperature. So it's not at all obvious, to me at least, how temperature would affect these complicated biochemical networks and the behavior and the developmental rate of organisms. And so this is why in this talk I want to focus here on the biochemical network part and try to get some mechanistic understanding about how temperature affects these networks and the developmental rate of organisms. Okay. And I think we can start with the simplest thing, a single chemical reaction, which we're likely all familiar with, and it's described by a well-known Arrhenius equation. In other words, the reaction rate scales exponentially with the temperature. You can also plot at the reaction time in logarithmic scale versus the inverse of temperature, and in that case, what you get is a linear curve. So these are Arrhenius plots, and you expect to get a linear curve where the slope is the activation energy. Right? So this is for a single chemical reaction. However, if we look at more complex processes, such as developmental times, for instance, often this is still approximated by an Arrhenius equation, and there's no clear reason why this would be the case for such a complex system. So to give you an example, here I'm showing you a movie of the early embryonic cleavages, the earliest divisions in the frog Xenopus laevis, in the embryo. And as you can see, they divide well, you can't see that it's quickly, but they divide every 20, 30 minutes or so. And all of them divide at the same time very quickly and in a clock-like manner. And so if you analyze the period of those cell cleavages, and that's what's shown here, in function of the inverse of temperature, this is no longer a straight line. It's curved all the way through over the entire temperature range. So what we're interested in is understanding why this is the case. And a nice thing about these frogs that I just showed is that you can also look at the system in vitro. So these early cleavages, they're basically driven, they're very clock-like, they're basically driven by an underlying biochemical oscillator with a certain period. And you can reconstitute that oscillator using cell-free extract. So basically, you mix all the frog eggs, you extract the cytoplasmic fraction, and then we use these extracts to put them in droplets, encapsulate them in oil, and you can see that they flicker, they oscillate in time with a similar period as the embryos. You can see that under the microscope. You can also analyze it if you use an actual sensor for CDK1 activity, and CDK1 is the enzyme, cyclin-dependent kinase 1, that drives the cell division cycle. And so here you can nicely see the shape of the oscillator. So it allows us to look at the period and the shape of this biochemical oscillator that drives these oscillations. So using this system, we worked together a few years ago. We started together with Jan Rombouts in my group and Franco Tavella in the lab of Chong Yang at the University of Michigan, and together with Jim Farrell at Stanford University. We looked at this system, and what you can see here is CDK1 activity in function of time for 3 different temperatures. So we have hundreds of droplets at many different temperatures, and here I'm showing time series of just 3 droplets for 3 different temperatures. So here's an intermediate temperature, and not surprisingly, when you decrease the temperature, it becomes colder, the oscillator slows down. But when it becomes warmer, the oscillator also slows down, and that's perhaps a bit counterintuitive, right? So it means that in between, you sort of have an optimal temperature and optimal operating range. Maybe the previous then. So what we did then for these time series is we looked at each time series and we calculated the rising phase of CDK1 activity, the falling phase, and the total period. And we did that for these hundreds of droplets for all of these different temperatures. And here you can see the median of all of these results, where the duration is shown in function of the inverse of temperature. The blue is the total period, and you can see this U-shaped response curve. And this U-shaped response curve, you can also appreciate that it's mainly driven by the rising phase, which has a similar profile. Okay, to try to understand why this is the case, we turn to modeling, and we use a very simple cell cycle model where basically we're only looking at how cyclins, these are synthesized and how they're degraded, and how cyclin-dependent kinase, together with cyclins, the protein complex is activated and inactivated. And this leads to 2 differential equations, which you don't really need to pay attention to, but I just want to show you that's 2 differential equations. And you can simulate that, and you can get a pretty good model for the experimental time series that you measure. Now, the nice thing about having such a model is that we can now try and see how the parameters affect the dynamics, and we introduce a temperature dependence into each of these 4 parameters, the 4 reaction rates, and then we use that to fit the data that I just showed you of these rising times, falling times, total periods, and you can see here the 100 best fits, and they sort of all match the data pretty well. So the model does a pretty good job, despite it being quite simple. And the nice thing of that is that now we have this optimal fit, but we also now have the temperature response of the optimal solutions for the underlying reaction rates and how they respond to temperature. And so that's what I'm showing here. You've got the cyclin synthesis rate in function of temperature and the degradation rate. And what you can see is that the synthesis rate has a biphasic response. It speeds up when the temperature goes up, and then it goes down. And the cyclin degradation rate does not change a lot with temperature. So you've got a different temperature sensitivity between the synthesis rate and the degradation rate as well. Right? And so it turns out that both of these features are important to explain how the total temperature response changes. Okay, so but not all processes are cyclic, not all driven are by simple periodic oscillators. You also have more complicated developmental events. And so together with Simon Jacobs and Nikita Frolov in our group and with Federico Vasquez, we looked at a system that essentially is going from one state to the next through a chain of multiple steps, where you can go forward and you can go backward. And in all of these different transition rates, we include also this Arrhenius scaling to have a temperature dependence. And the scaling for each individual step is drawn from a distribution where you have activation energies drawn from a certain distribution. So what you get is all of these forwards transition times form a distribution, the backwards ones as well. And so this distribution, we shifted a bit so that there's directionality for the developmental process. So on average, the forward steps are faster than the backward steps, right? So using this, we get to a sort of solution that is like a very stereotypical scaling profile where you have the time to go through the whole process versus temperature. And what we see is that on the cold side, you scale Arrhenius. Basically, one process is going to dominate. In the middle, or I'll start on the warm side, you also have Arrhenius scaling. And what is happening there is that instead of this long cascade, you've got certain cycles that start dominating. You go forward and backwards, and you get stuck at certain locations. And this gives a negative activation energy. And then in the middle for this temperature range, you've got sort of collective behavior where you average all of these many steps, and you have a quadratic exponential scaling, which is essentially also what Julian Voits and Ulrich Schwarz found in their beautiful paper last year. So this is the standard scaling, I will finish now, that we find. And so what we did then is we simulated this over many different realizations, many different simulations, all new drawings from the distributions. And what you can see is that in the quadratic range, everything clusters very nicely together, while at the cold and the warm side, there's a lot of variability. So not only do you slow down there, but it also becomes very unpredictable. Whereas in this middle range, you're fast and robust in your sort of transition timing. Okay, so then this is my last slide. We wanted to check whether there's data sets to test these observations or these predictions. And so here I'm showing for Xenopus laevis the duration of early cleavages again, and it's perfectly fitted by a quadratic exponential. Here I'm showing just a single cell green algae, the cell growth, where we found a lot of data. And so here this dashed line is the quadratic, and you see that it deviates at the cold side, right? But again, perfect fit. Then for C. brixae, small nematodes, now it's the opposite. So here you've got the fit of the quadratic, and then you deviate at the warm side. And then here another example, Xenopus tropicalis, so another frog, and here it deviates at both the warm and the cold side. But the fit is always really, really good. Another thing that is nice to see is that when you deviate from the quadratic, here these error bars go up. So just like the theory says, like whenever you deviate from the quadratic scaling, everything becomes very variable. And we did this for around 50 data sets where we had a lot of temperature resolution. And many of them scale quadratically. Also many scale quadratic and deviate at the warm side. And then you have these two other cases which are a bit less prominent there. And so I think, I want to end with that and thank all the people that were involved in both parts of this presentation. And there are two people here, Jan and Nikita, who are in the meeting, so also visit their posters. Thank you.